Hi, good afternoon, everyone. So I want to spend the next 20 minutes talking a little bit about growing food with electricity and how you can do it at your kitchen table with a little open source bioreactor. So I'm presenting this as part of Amiibo, which is a online community focusing on development of sustainable protein for everyone. So who am I? I'm the co-founder of LabCrafter, a small hardware company here in the UK that sells open source scientific equipment. So things like microscopes, calorimeters, bioreactors, and maybe even one day the moth box, who knows. In the past, I've designed a open source syringe pump, a hydroponics controller, and an outdoor air quality monitor. And when I was working on the outdoor air quality monitor, I developed a little breakout board, a little 5-volt boost converter that's really useful for driving 5-volt sensors like air quality, particulate matter sensors. And what's nice about this little board is that it has true shutdown. So you can turn it off with a microcontroller, meaning that when it's turned off, your sensor is completely turned off and it doesn't drain any additional power. So very useful if you're building something battery powered, such as a outdoor air quality monitor. So I got that certified by OSHWA as UK 43. So I've got my OSHWA certification. Andy actually already made a very good concrete example of what I want to tell you now, and this is land use. So all these different colors here on this graph indicate all the land on Earth. So the blue area is barren land and glaciers, so non-habitable land, and everything else is habitable land. And then if you look at the orange block right in the top left, that is the amount of habitable land that is currently used for livestock. So it's about 40 million square kilometers. 32 million square kilometers is used for grazing. The other 8 million square kilometers is used for growing the food to feed to the animals. The little other orange block just below there is the amount of land that we need to grow all the plants that we feed to humans. So about a third of the habitable land on Earth is just being used for livestock, which is our main source of protein at the moment. So this is also, industrial agriculture is also responsible for around 15% of global greenhouse gas emissions. So this is something where we can obviously do better, where we need to find alternative sustainable sources of protein. And one of the things that we've been looking at Amiibo is biomass fermentation. So biomass fermentation, the example that most of you are probably familiar with is this green stuff here called spirulina, where this gets grown in big outdoor raceway ponds. So it photosynthesizes, so it's relatively easy to grow. You can turn it into a green powder, but it's not really been able to become an alternative source of protein. First of all, because it doesn't really taste that nice. It has a bit of a fishy taste to it. We usually eat it in quite small amounts as like a supplement. So you'll turn it into, it gets turned into like like tablets maybe, or you'll have it maybe added to your smoothie, or at some of the supermarkets you can buy these health shots that have spirulina in them. But it is something that I think is probably the best example of biomass fermentation, and it is a really good source of protein. Unfortunately, there's some issues with toxins when you try to produce this in large amounts. So at the moment, we really only consume these in quite small quantities. This here is corn with a Q. It's quite popular here in the UK, I think in the US as well. It is a fungus called Fusarium venenatum. And it is based on a technology that got created in the 80s, where they started building these massive stainless steel bioreactors, growing these fungi inside the bioreactors. But it is a very sustainable source of protein, at least a lot more sustainable than animal protein at the moment. It is also very versatile, so it gets turned into basically everything. I mean, if you walk into a supermarket here in the UK and you go to the aisle where they sell corn, you can buy something that resembles almost any kind of animal protein. One potential issue with this is that you still need to feed something to the fungus. So in this case, it is glucose. So we get the glucose from either wheat or from maize. So at the end of the day, you still need quite a lot of land to grow the wheat and the maize to turn into glucose to then feed to the fungi, which you can then turn into food for humans. So a couple of years ago, I started hearing about this thing called air protein, where you're essentially creating protein out of thin air. Now, it turns out that, like all things, it's not really creating it out of thin air. It's actually, I would say, creating it using electricity. Because what you're doing is you're taking electricity and you're splitting water into hydrogen and oxygen. You're then feeding that hydrogen and oxygen and carbon dioxide to a bacteria called the hydrogen-oxidizing bacteria. And this bacteria is then what you harvest and what you can then turn into edible protein. So basically your source, your feedstock is all these different gases, which is why it's then called air protein, because these are gases that exist in the atmosphere, but we're not feeding the atmosphere to the bacteria. You have to create the hydrogen and feed the hydrogen and the carbon dioxide to these bacteria. The carbon dioxide can either come from existing sources that generate it, like, I don't know, beer breweries, but maybe in future we can capture it from the air. At the moment, it doesn't seem that technology is going anywhere, but maybe in future it will. So these are the commercial companies currently in the space that are working on developing this kind of protein. You've got Air Protein in the US, you've got Farmless in the Netherlands, you've got Solar Foods out of Finland, Nova Nutrients, Callista. AirBio is a company that previously used to be called Deep Branch. They were based here in the UK. I think they then moved out to the Netherlands, and then currently they're based in Denmark. I would say the market leader in this field is Solar Foods. Some of you may already have heard of them. They have novel food authorization in Singapore, and they're also cleared for use in the US. Basically, what novel food authorization is, if there is a food source that hasn't been consumed by humans up until very recently, it needs to go through a regulatory process to make sure that it is safe before you can put it on the market and sell it to for people to eat. So Solar Foods hasn't been cleared for use in the UK or Europe yet, but yeah, they have been selling their version of this hydrogen oxidizing bacteria protein in Singapore, and I think very soon in the US as well. This is what it looks like. So it also gets turned into a powder. It's kind of golden yellowish powder. It is a lot more of a neutral taste, a bit of a umami taste, which means that it can get turned into a lot of products. It's a lot closer in that sense to corn than what it is to spirulina. So they've turned it into ice cream. They've turned it into pasta. So yeah, there's a lot of very wide variety of foods that this can be kind of turned into as a protein source. This here is Solar Foods bioreactor. Again, a big stainless steel bioreactor that they then use to grow these bacteria in, feeding them the hydrogen and carbon dioxide. But there's one problem with all of this, and this is that all these existing efforts are closed source and proprietary. Most of the technology is patented, and I would like to play with this at home. I would like to grow my own. I mean, we do that with other food, right? Most other foods are not locked down, and I would love to do this myself. So as part of Amiibo, we started looking in how can we do this open source. As soon as I found out about this, I was like Googling, like, oh, is there any kind of people working on this in open source? And it turns out there was nobody. So that's why we kind of started Amiibo, to be able to try and get people together to see if we can do this the open source way. I also came across this manifesto, the Reboot food manifesto that was written by a group called We Planet. And they kind of had these 4 principles where they're like, we need to make things plant-centered. We should brew, don't slaughter. What they mean with this is you get this a technology called precision fermentation, where essentially you can genetically engineer yeasts or bacteria to create proteins, like let's say whey protein or casein, so that you can then produce milk, turn that into cheese, so you can have real vegan cheese. So that's what they mean with brew, don't slaughter. The idea that we can rather use microorganisms like yeast and bacteria to grow all the proteins and different foods that we need. Also, the third principle, then use as little land as possible, rewild everything else. We've already seen quite a few examples of why this is necessary. And then I really thought the last one was very interesting, where they said open source everything. New technologies in food should be open source, and corporate concentration must be actively mitigated to ensure the benefits of the food revolution are shared equally with all, which I think was a really nice, succinct way of saying like why this should be open source. So we got started, and the first thing that we needed to find was a open source bioreactor, and we came across this. It's called the pioreactor. It's a Raspberry Pi based bioreactor. It is used to grow, monitor, and control microbial cultures. So there's a little 20 milliliter vial inside. There's also now a larger 40 milliliter version. And then you can grow your bacteria inside this vial. There on the sides, you've got infrared diodes and photodiodes, which allows you to do optical density measurements. This allows you to measure how quickly your culture is growing. So you can figure out how fast everything is growing inside the vial. There's liquid handling. So it has peristaltic pumps where you can either add new culture media to your vial or you can remove the existing protein that you're growing out of the vial. It has temperature control. So there's like a little temperature board that sits in the bottom that keeps everything at a constant temperature. And then, of course, because this is open source, you can add your own sensors and actuators. So we've added little spectrometer boards to it. You can add relays to it. If you do want to grow spirulina in it, you can. You can add LEDs to it, which you can automate to turn on and off at certain times. So it's quite a versatile platform to get started with kind of like developing different types of microorganisms in a lab, in a home lab. So this is the setup that we came up with. We started off by saying, okay, how are we going to do the carbon dioxide? Easiest thing was just to get a SodaStream cylinder. Those are quite easy to obtain. They're easy to refill. And then, so that's how we put the carbon dioxide into the system. We then also needed a way of generating the hydrogen and oxygen. So we thought, okay, how about instead of having hydrogen gas canisters, which is obviously quite unsafe, maybe unsafe, how about we generate the hydrogen and oxygen inside the vial itself? So we put little electrodes in there, supply electricity, and we can generate hydrogen and oxygen right inside the vial itself. We're also then using the pioreactor to do the optical density measurements, spectrometer readings, and keep the temperature at a constant level. When I started off with looking for electrodes, I got a little platinum-coated titanium electrode off eBay, cut it up into a smaller piece that would fit inside the vial, started up the electrolysis process, and I ended up with a lot of sediment inside my vial and realized that there was a lot of oxidation that was occurring. And obviously, when you buy something with platinum in it off eBay, it's not necessarily platinum. So we ended up finding a more reputable source of platinum-coated titanium electrodes. And this was my second experiment where things looked a lot better. I was able to run it for hours, days, and it would just generate the hydrogen and oxygen inside the vial with no sediment forming, which was great. So we proved at least that our electrolysis is working. For carbon dioxide sparging, I mentioned the SodaStream cylinder. We connected a regulator to the SodaStream cylinder. with a little solenoid valve so that we can then control it from the bioreactor. So we can set up an automation that says every hour, sparge 10 seconds, and then sparge the carbon dioxide into the vial from there. Got a couple of non-return valves on there as well, so that to try and prevent backflow out of the vial into the tubing. We're working on various improvements for that. So maybe we're working trying a pinch valve instead of the solenoid valve, or moving the solenoid valve closer to the vial itself instead of it sitting on top of the regulator. And this is what the setup looks like. So on the left, you've got the bioreactor. Next to it, you've got the peristaltic pumps that then receive new media from the centrifuge tubes on the right-hand side, and can then also remove the growing protein from the bioreactor into the tubes on the right as well. And then we've got the soda stream cylinder there as well. So just a quick note on mixed cultures versus pure cultures. So usually when you work in a lab, you're working with pure cultures. If you're working at home, you may not have the same sterile environments and aseptic techniques that people in a lab may have. So that makes it a little bit more difficult to work with these pure cultures. They can also be quite expensive. So we went for the alternative of looking at mixed cultures, meaning that what you do is you go and collect samples from the environment in areas where you think hydrogen oxidizing bacteria may exist. This can be in a mud flat near the sea, for example. You then put that sample inside your system and you start enriching your sample. Hopefully, because you're providing just the environment for your hydrogen oxidizing bacteria to grow, you're not growing other stuff, you're just growing what you're hoping to grow. And then you end up with a mixed culture of different species of hydrogen oxidizing bacteria instead of a single species or pure culture, what people typically work with. So the reason why this is not done in industry is because it is extremely difficult, if not impossible, to get a mixed culture through the regulatory process. They're really focused on pure cultures, but maybe the regulatory process will change in future, and we will be able to have mixed cultures go through that process as well. Right, so here is me on the left, taking some samples in a forest near my house. On the right there, we've got Martin, one of our other volunteers and founders. He went to Glencoe and collected some samples there. And that's kind of the stage where we are at the moment, where we're hoping to get more people to replicate our setup, go out and collect samples, and see if we can grow hydrogen oxidizing bacteria. Yeah, so please come and join us. We've got our website is over at amybo.org. We've got a forum at forum.amybo.org. We've got our GitHub up. And then LabCrafter is my own company where we are selling pioreactors. And hopefully soon in the near future, we'll be selling an add-on kit for growing hydrogen oxidizing bacteria as well. Great, thank you.